Good evening, everyone, and welcome to tonight's very special program. My name is Don Morell, and I'm the chair of the Board of Trustees at the Franklin Institute. This week, we kick off our Awards Week activities to celebrate the achievements of our class of 2021 Franklin Science Laureates. We're particularly pleased this year that we can celebrate this, albeit virtually. There's been few years like 2020 where science and the power of science has been in such public view, affecting all of us around the globe. Tonight's program is very special, and I will kick off with a thank you to Dr. Darrell Williams, who heads our science programs groups and science education, and Beth Sharaga, who's our director of the awards program, as well as Professor Alan Nicholson, who heads the Committee on the Science and the Arts. We are having our inaugural NextGen Science Award tonight. It is one of the first new awards since the Franklin Laureates were begun to be recognized as far back as 1824. We're almost 200 years old now with the awards program, and we have been fortunate to honor the luminaries of science over that time period. Tonight, it's my distinct honor to welcome Dr. Kizzmekia Corbett of the National Institute of Allergy and Infectious Diseases, who is our inaugural NextGen Award winner. And as you'll see in just a moment, this is appropriate in 2020, given the spread of the novel SARS-CoV-2 coronavirus and her work as it relates to immunology and vaccines, which is quite fascinating. The purpose of the NextGen Award is to bring the public's attention to the work of younger scientists. We typically honor those for career achievements, but given our mission of inspiring a passion for learning about science and technology, we thought it appropriate and probably beyond time to recognize younger scientists as role models for the next generation of scientists coming forward. Dr. Corbett is currently Senior Research Fellow and Scientific Lead at the National Institutes of Allergy and Infectious Diseases and the Vaccine Research Center. Her journey is a fascinating one. This evening, we're going to spend a conversation with her about that journey and about some of her scientific work. We're hoping that this conversation will inspire a number of young scientists to follow in her footsteps. We are very fortunate that Dr. Gayatri Das, who is our lead bioscientist at the Institute, has agreed to take on the rather daunting task of providing the focus for the conversation. She'll be joined by a very special guest, Talid Hamad. We're welcome, Talid. Very nice to have you join us this evening. With that, I will turn the program over to Gayatri and offer my congratulations to Dr. Corbett again for being our inaugural NextGen Science Award winner. So with that, Gayatri, I turn the program over to you.
Thanks so much, Don. Before we jump into our conversation tonight, I have a couple of housekeeping notes about our Zoom interface tonight. Those of you in the audience, you'll notice that there are options both for the chat and Q&A. And we welcome you to share your experiences and thoughts with us in the chat box. But if you have a specific question for Dr. Corbett, please go ahead and put those in the Q&A, and we'll try and work those into our conversation as much as we can. It's a true honor for us to be joined by Dr. Corbett tonight because she's such a powerful voice for science at this moment. Over the last year, we've seen public health come into focus through the intertwined challenges of both the emerging pandemic on one hand and the ongoing impacts of systemic racism on the other. Dr. Corbett, your leadership in the lab and in the community demonstrate how we can tackle these challenges by bringing diverse perspectives and people together. It's been so much fun for me to work with the young scientists in our STEM Scholars program as we've learned about you and your work over the last few weeks. So I'm going to actually turn it over to Talid to kick off our conversation tonight.
Yeah, I agree entirely with Dr. Das. Learning about Dr. Corbett has been an amazing experience. Every note I've read has brought me more faith in my own ability to succeed as a woman in STEM. And Dr. Corbett's work with the corona vaccines, especially in such a short amount of time, shows how truly dedicated she is to her work. And I know I and many other students in STEM admire her for it and want to reflect that in our own work. As all dedication must come from true inspiration, Dr. Corbett, I was wondering what inspired and led you to join the NIH team, and when did the NIH team begin researching coronaviruses?
firstly, thank you so much for the invitation. And of course, thank you for this extremely, extremely prestigious honor. I am thrilled to be here. And I'm thrilled to have this conversation because all the things that the Franklin Institute stands for are things that I actually hold very dear to my heart. So thank you again. The NIH and my place at the NIH is actually one that is a little bit odd, because when I was in undergrad at the University of Maryland, Baltimore County, I was a Meyerhoff Scholar, but also I was a NIH undergraduate scholar. So they have an undergraduate scholarship program, or so-called UGSP, where they give you $20,000 a year to study at your respective university. But then after each year, you are able to come to the NIH and do an internship in the summer. And so I was exposed to what was government research at the NIH very early when I was 19 years old. And I loved it. I loved it there. And because I was in the UGSP scholarship program, I was able to come back as a research fellow. And so they actually, aside from the internships, afford you the opportunity to come back each year as a research fellow. So if anyone is out there and they're an undergrad and they really see themselves as a career scientist, that type of program would probably behoove you if you're really interested in working at the NIH, because it is a step into the door. As far as the team that I work on, it's interesting because I work with Dr. Barney Graham, or the Vaccinator is what the world calls him, and he's a career vaccinologist. And I wanted to do vaccine research because I think it sits at this interface of viral immunology, which is my expertise by way of my PhD training, but then also health disparities in an interesting way, because vaccines have the ability to really level the playing field around health disparities. And so I approached Dr. Graham around working in his laboratory. And I have to ask him at some point whether he allowed me to lead the team because he just believed in me, or because coronaviruses, would not necessarily one of the largest product portfolios at the Vaccine Research Center. But nevertheless, I really kind of stumbled upon being a team lead in that way. I was really interested in having a research niche that would allow me to become a principal investigator one day and carry forth something that was external to the VRC that I could maybe move into academia with. And so coronaviruses seemed like it would be a good fit. And I think all things essentially worked out for the good in that way.
So I think one of the reasons that the NIH exists is to, tackle these big challenges that are facing our country's health. And yet coronaviruses, weren't necessarily on the tip of everyone's tongue a year ago. So how did coronaviruses get on the NIH's radar?
The NIH actually is that kind of place where, because there are two sides to the NIH. So there is the extramural side of things where any investigator in the United States can apply for a grant and get funding. That's actually the biggest part of the NIH. And then there is intramural, which is internal, like the center that I work in, or all of the other, I think 24 institutes and centers at the NIH. Please forgive me if that number is incorrect, but we have this really unique ability intramurally to work on things that are rare questions in science that are understudied, generally speaking, because that's what the resources are for. High risk, high reward type projects. And that's essentially what coronaviruses are. We knew by way of history that coronaviruses had the potential to cause a pandemic. So SARS had circulated over a decade ago. MERS, actually, when I started in 2014, was circulating, and actually still steady state circulates in the Middle East. And so it was only by chance and luck that we hadn't seen this type of pandemic spread with the coronavirus, but we knew that it possibly could exist. And so this portfolio of research where coronaviruses sit, Zika virus, Nipah virus, and other viruses like that, even pandemic influenza viruses, is called the Pandemic Preparedness portfolio. And it's a large focus at the Vaccine Research Center because you want to be able to do this type of thing, really move very quickly by way of vaccine development, shall a pandemic occur with any of the, I guess, 24 viral families that have pandemic potential.
I know you studied with both immunology and sociology. How has your background in each field enhanced your understanding of the other?
Yeah, so in undergrad, I did biology and sociology, and I actually did I did sociology because my first sociology professor, Dr. Kelly Moore, who's now at Case Western, but was then at the University of Maryland, Baltimore County, was just amazing. And she was working on this project called the Handles Project, which is healthy aging in neighborhoods of diversity across a lifespan. So it's this very long-term project where you look at what are neighborhood effects on people's health. And being in Baltimore is a really interesting place in that way, because you can really see how in one neighborhood versus over the next street over, people's health outcomes are drastically different. And mostly because of what are the tools that are available to them, whether it be grocery stores and healthy eating and you know, doctors, et cetera. And so for me, that really stuck with me this entire time. And I think that one of the things that came out of this pandemic is that there are certain communities that were just disproportionately affected. You know, African Americans had at one point even 4 times more deaths than their white counterparts. And so health disparities exist, and they always will until we fix them. Vaccines are certainly not the only way. There are structural determinants of health disparities that I don't need to go into. But from where I sit as a viral immunologist, the least I could do is do vaccine development to try to level the playing field as much as I could. And so I thought that that was how the two fields really came together. And by no means, I'm not a sociology expert. I wish I wish I could claim that, but I do, I think, have a fairly intricate understanding of health disparities, and that helps me a lot.
You know, I think that that's such an important point that you make, that, you know, whether it's the, you know, studying different fields, but also your lived experience comes into play as your perspective as a scientist. And I, you know, you've talked a lot about how that's shaped your perspective. And I thought maybe you could tell us a little bit about that personal perspective before we get into more of your science. So what's your experience been like as an African-American woman and a scientist in STEM? What opportunities does that bring you, and what are the challenges as we think about this, you know, this challenge that we're seeing that does have such disparate effects on our communities?
Well, one thing that really stands out to me in this moment, I feel like I may have been in a bubble around what is the visibility of African-American scientists. And actually the first scientist that I ever met in my life, the first PhD scientist that I ever met in my life, was an African-American man. His name's Albert Russell, and I interned with him when I was in high school. And so I may have had what is a cloudy view of what is the landscape of science, which is predominantly white male, and that is okay. What has been important to me along the way is that I continue to stay true to who I am. I really think that everyone's place in society really overlays with what is their place in the workplace. And so I show up as Kizzmekia Corbett, Black girl from North Carolina, in every room that I enter, and I allow my data to speak for me. And I try to do that as much as possible. And I try to also teach that to students that we have in the laboratory as well, because there becomes a time where your seat at the table is yours to own, and figuring out how to sit in that seat actually is always a question for anyone who doesn't necessarily see people like them at the table. And so, you know, for me, the experience has been one that has continuously been a learning experience. You figure out your way. I think that we certainly have a very long way to go. There is a movement towards diversity and equity and inclusion. The NIH just released Unite, which is a policy around racial inequities at the National Institutes of Health. And so there's certainly this understanding that something has to be done. But I think what is the key piece is the belonging piece, where everyone at the table feels like they belong there. And I just have to give credit to my boss, honestly, because he really has curated this table where every single person has a voice and that their voice is heard. He makes it a point to, when he's asked a question, to defer if he feels like someone that is not in the leadership position could answer the question equally as well as him. He makes it a point to, I mean, in the beginning of the pandemic, he literally sat me down and he said, we need to figure out what we're going to do. We had about a 2-hour meeting. And as I was leaving, he said, I'm going to push you to your limit because this is your time. And just let me know if it ever becomes too much for you. And so the next day I was included on WHO calls and, CDC, all of these, meeting presidents. And so that type of atmosphere where, because someone had seen what my potential was, they carved out a space for me. And I don't necessarily know that without him doing that, I would even be talking to you, honestly. And so I think that it's important for people to just realize or remember that, find that person or those people and be grateful for them, number one, but then also let them guide you. I think that he and our long relationship, because I met him when I was in high school, has gotten in tune with how much I can take and where he needs to step in versus when he doesn't. And that's been very helpful.
Thank you so much for sharing that experience with us. Now that we've gotten to know a little more about you, can you tell us about can you take us behind the scenes at NIH back in January 2020? What was happening as you learned about this new virus, and how did you determine your approach to the vaccine?
Oh man. Okay. So I'll take you to December 31st, 2019. I got an email from Dr. Barney Graham, who was the boss that I just spoke about. And it was an alert about what was the respiratory virus that was then circulating in the Wuhan district of China. And it was, it was just be ready for 2020. And we didn't necessarily know what that means. Honestly, I wrote it off. Okay, we have an avian flu. Okay, I won't be in charge of that. I don't lead the flu team. But when we got back and we realized that it was the coronavirus, we really set into a plan of action. And frankly, in our portfolio of platforms, we tested our antigen. So what an antigen is the thing that is delivered via the vaccine. So in our case, it's a protein from the coronavirus that we modified to make it a good thing for a vaccine. And we tested it in all types of platforms, but messenger RNA is a platform that obviously works very well. But then also you can manufacture it quickly enough that you can maybe beat a pandemic spread of a virus. And so that was extremely, extremely important as we thought about which type of technology to use. And so we just set a plan into action, and everybody on the team had what was the thing that they did. I think I like to say that it was a McDonald's moment. We really had an assembly line of, okay, the sequence comes out, who downloads the sequence, who aligns it, who checks it, who orders mice. We had an entire thing ready to go. We had mice ordered, ready to go, even before the sequence came out, materials ordered. And so it was really, this classical preparedness situation that we had spent the last 6 years preparing for.
So that's really fascinating that, the rest of the world, at least it felt like, got caught by surprise. And yet, you were in this team that knew exactly what had to happen.
Well, I don't know if we knew exactly what needed to happen. I think that we knew how we could best serve the situation, because there are certainly several other things that needed to happen that we weren't expert in, right? Like, PPE or therapeutics and all of these things. And so I would say that we're also very caught by surprise. I took my career in this direction, and I went to the Vaccine Research Center so that I could observe what was translational research. But I never would have in a million years thought, especially by 35, that I would be, writing documents to support a phase 1 clinical trial for any vaccine product, frankly. And so I think that there is the surprise element that still remains the same for us as well as it did for probably everyone in the world. But I think that the difference is that we had a system that we believed in.
Right. So take us through that system a little bit, because we've heard so much about, the human clinical trials as, these vaccines get to the late stages. But the early work that you led first starts in animals. So what does that animal testing look like, and what strategies did you take?
The interesting thing is that we had tested MERS, which is a cousin of the SARS-CoV-2 virus that causes COVID-19 vaccines, and delivered them by mRNA before. And so we'd already done a plethora of mouse experiments, many of which will never be published because they ended up being, one supplement figure with 2 panels in that Nature paper. But we understood dosing for protection and also subprotection. We knew Dr. Graham, because he has built his career on respiratory syncytial virus, we kind of understood what could be the potential for vaccine-induced enhancement. So that's a phenomenon where a vaccine is not as helpful as you would like it to be, but instead might cause actually disease. And so we knew how to test for that even in the preclinical setting or in the animal models. And so we designed experiments to test dosing. We had this massive portfolio of collaborators. I graduated from the United States, from the University of North Carolina at Chapel Hill, where Ralph Barrick's lab is. I like to think of him as the OG or the original gangster of coronaviruses. He's done coronavirus research all of his life, and he is a master in animal models. And we collaborated with them so that we would be able to use their animal models first. So all of these things came into play around making sure that as quickly as possible, we could go into a phase 1 clinical trial. And the experiments that inform phase 1 clinical trials are really, does this vaccine, well, number one, is it safe in animals, but then also does it, elicit the types of immune responses that you can foresee being immunogenic in a person? And to answer those questions very quickly, we really took advantage of all of the knowledge that we had curated over the last several years.
We have a quick audience question for you. How did working on the vaccine challenge and your personal strengths and how did working on the vaccine challenge your personal strengths and capabilities, and in what ways did it change you as a person?
Oh man. So I've always been fairly churchy. I'm Southern Baptist, spent at least 2 nights a week in church, growing up, and I really relied a lot on my faith throughout this entire process. I got criticized very heavily for my first national appearance on CNN with Anderson Cooper. And I was like, oh yeah, we'll have a vaccine by fall. And everyone's like, what? But when you allow the universe to tell you what are your strengths, I think you fold into that, and it really helps a lot. So I've obviously been tested in so many different directions. My sleep is massively deprived. I'm actually on an all-nighter right now because we're submitting a paper tomorrow. But I have really leaned a lot on not just my faith and my pastor, but also my family, my friends, my partner. It's just really helpful to have a support system. And then again, Dr. Graham is the best at just recognizing what are my limits and just saying, okay, I'll take this. I can't read this discussion anymore. And I left 3 of your comments there. Just go polish the sentences. I can't do it. And he's okay with that. And so realizing where your breaking point is very, very important in this type of moment. I think at this point, the world is tired. We're doing research, but then also at the same time, we're still living in a pandemic. And so recognizing that, And, you know, and finding the type of support that you need is important. And that's not just for a pandemic moment. The lesson of it all, for me, at least, is I've learned how to say no more than I ever thought that I would ever have to. It's a blessing to be asked for things from basically every corner of the globe at this point, but you have to be able to say no. And so I have, you know, a system where you know, if I give this talk tonight or talk to you guys tonight, then I don't talk tomorrow, even though I am. But anyway, I try not to talk 2 nights in a row. I try to space things out. I try to make sure I balance between what's academic talks, which can be a little bit more stressful, frankly, than in community talks. And so you learn where your boundaries are.
I think that's such an important point that, you know, we're all human. And I think this crisis has really taught us how we depend on each other in so many ways that you know, we may not have realized before. And certainly even just the way that vaccines work is something that, you know, asks us to build that community with each other. So, you know, a lot of the after hours work that you do is really focused on getting people to understand the science that you do. Why do you think that helps people to understand that science? And how do you actually figure out how to explain the science that can be pretty complicated sometimes?
Well, I mean, at first, I feel like I owe everybody a sincere apology that when we came into this moment, we had these patents and we had these manuscripts, but nobody in the general public knew that. And I was in the back of this laboratory leading this 4-person team, busting out what are, you know, from a science perspective, great data, but just not speaking about it. And so no one knew what was to come. And that is only the fault of me and us. And so apologies for that. And so this moment really made me realize that you're not really doing that much good if you're not making sure that people understand the science and you're not being tender and empathetic to the people who don't have the privilege to call Dr. Fauci and say, wait, they're blood clots, what's going on? Like, you know, and so I think that for me, this moment has really opened my eyes to that. And I found that it's just so important for us as scientists to really learn as soon as possible to pare down what is our academic talk to more lay audiences. And so I try so very hard to make sure that I do that as much as possible. And I mean, I have a speech coach who helps me, and I took the onus on myself, and I am going to try very hard, first of all, to pray very hard with everyone that there's never another pandemic, at least in our lifetime. But if there is, I want people to know that there are 24 viral families that some lab around the world is working on a pandemic preparedness plan for, and that science needs to be spoken about more often than not to the community.
Going off of that, we have another audience question for you. How are vaccines tested for safety, and how do you communicate their safety?
There are several different layers to testing vaccine safety, much of which, again, is just this big gray space for the general public. And so the first layer that I spoke about was simply testing in animals. And you can find context clues in the data that you get from animals. Their immune responses, certain types of adverse events can be correlated back to animals' protein responses that they have from their injections. Then the first level of a clinical trial, the phase 1 clinical trial, the endpoint. So that's the thing that the FDA looks for the most that determines whether you can go to your next phase of clinical trial is actually safety. And the way that that's tested is by testing, usually speaking, or generally speaking, a range of doses of the particular vaccine in humans, and then assessing people very consistently in that trial in about 50 people and looking for, fever, all of the things that are considered to be adverse events. And those things are taken extremely seriously. And before you can go into the next phase of your clinical trial, you have to prove beyond a shadow of a doubt that the vaccine would be safe for the next stage, which is about 500 people of testing. And at each stage, while the data that you hear about when you watch the news, or even when I give my talks, frankly, because I don't really talk about the safety data, but at each stage, safety is the priority. It is always the top priority, especially with vaccines, where what you're giving to people, you're giving it to, generally speaking, healthy people with the idea that they might come in contact with the virus later. So whatever comes out of that product has to be something that is so safe that the risk assessment means that if they get this vaccine, they will by far fare much better than they would with the possibility of getting the disease.
I think that's such an important point in helping people understand the risks and benefits. And it's a challenge, right? Because you're asking people to think of a hypothetical in many cases when it comes
to, I think about that. I think about that hypothetical, and I think that, I don't know if you guys watch what are called the VRBPAC, excuse me if I'm saying that wrong, but it's essentially the advisory committee to the FDA, where they hold these public meetings and they basically look at all of the data. And at the end, the entire meeting is about like the risk assessment. So it's not as easy as it looks. And there are people who are experts who do this on a day in and a day out basis. But yeah, there are two levels to the risk assessments, especially with vaccines. The first level is individual. If I, as a healthy vegetarian, dabble of seafood eater, prayed up woman, get coronavirus, the likelihood that I would be okay is very high. And so if I'm making a risk assessment about getting vaccinated for myself, then I might say, I don't want to get the vaccine because I'll just get coronavirus and it'll be okay. But when you think about it outside of yourself in your risk assessment, it really is about your community. It's about, if I get coronavirus, as I'm going about my day-to-day life, who could I potentially pass that to? Could that be an elderly person? Could that be someone who, has diabetes, or what other comorbidities are for the disease? And so when we think about vaccines, we like to think about risk assessment in that way. Also, we like to think about what is the efficiency of getting us out of the pandemic. And by far, one of the most efficient ways is vaccination.
So we've seen, I think, some really interesting shifts in people's willingness to take the vaccine, over the last few months as the rollout has happened. Certainly, you've spoken with so many African-American communities in particular who have a lot of reasons for being hesitant. And we see those shifts in the data, in polling data now. There are now other communities now that are more hesitant. What factors, do you see, playing into people's decision about getting the vaccine, and how do we bring everybody along?
Well, okay. So first thing I want to say is that I have largely spoken to African-American communities because that's where I've been invited. But I feel like my purpose extends beyond, and I'm very happy to talk to any vaccine inquisitive group, no matter who they are, because I think that it's important for my voice to be heard across barriers. And that's how I try to live my life, and definitely how I try to go about my outreach and service. One of the major things about vaccine inquisitiveness is that really what it is that most people just have questions. So we call it hesitant, but answering that question, or those questions that people have on a very individual basis, helps a lot. The other thing is that as these vaccines, multiple, especially in the United States, have been given to millions of people, and people are being told by, their grandmother or sister, brother, who've gotten the vaccine, and they are seeing that people are okay. that likely changes their mind a lot around the vaccine. I think most people just felt that the vaccines were so new, on top of the fact that there's this novel virus. It was a lot to take on initially. And frankly, even for me, I obviously would have gotten the vaccine on day one, but there's just so much information and hard to really dissect that from the general public standpoint. And so what is our role as scientists is to just guide people through that as best we can, and to remind people that of where the resources are, the trusted resources are, and where they should get their information, I think is the most important thing around that. And, the numbers are better, but the numbers are going to get to the point where you reach a threshold where you're not going to be able to successfully, and I don't like to say convince, or I like to say to successfully inform someone so that they make their own personal risk assessment towards getting the vaccine, is that there are just some people who are hesitant for their own reasons. And whether it be they have questions or whatever, it's just vaccines have gotten a bad name over the years. And so beyond the conversations that we have today and tomorrow with the COVID-19 vaccines, the conversations just have to continue. Whether it be infant vaccination or seasonal influenza vaccines or flu vaccines, we just have to keep those conversations open.
Seeing that the pandemic is a global challenge, what are your thoughts on the current state of the vaccine rollout around the world?
So my first thought, and has always been my thought, is I'm very thankful that I was on the side of development, and not distribution, because it is such a hard thing to even grasp, really. You have an entire world to vaccinate. The supply is, at least right now and for the foreseeable future, very limited. And so making those decisions, I empathize with the people or persons who are making those decisions. My thoughts are that we will get there. I think that one of the things that has come out of this entire effort scientifically is that people are really pulling together. Frankly, the CDC director in China graciously posted the sequences of the viruses, before publishing them even, because we knew that there was something that we all had to do, and we had to do it together. And I think that that's the way that we're going to be able to get out of this, that eventually there will become a point where vaccines are distributed to every single corner of the globe, and we'll get as many people vaccinated as possible. That day will not be tomorrow, unfortunately, but we will get there. And it becomes a waiting game and one of patience, I think, and just introspection around, this is what I can do in my place, and letting the experts do what it is that they have to do to make sure that the vaccines get to everyone. I had to come to that realization, because early on I was taking that as a burden and realizing I'm not even at that table, right? So I don't really necessarily have a voice at that table. I was taking it as a burden, like, oh my gosh, what if, for example, the African-American communities don't necessarily get prioritized or whatever? And so I just realized that that was not my burden to bear, and that I pass the torch in so many ways, but also pass the prayers to the people for which are handling that.
And on that note, I want to give a quick shout out to Dr. Ayla Stanford and the Black Doctors COVID Consortium here in Philadelphia, who have just, done incredible accomplishments to pick up that torch and get the vaccine out to folks.
There have been so many organizations along the way countless organizations that have just stepped up to the plate with their own expertise to make sure that this problem is solved in a very brick by brick fashion. Organizations stepping to the plate around diversity of recruitment into the clinical trials even. It's just been this massive collaborative effort beyond what I've ever seen. I'm obviously very young and I haven't seen much, but I'm hopeful that it just continues in this way all the time around therapeutics, vaccines, COVID-19, can't anything really.
And again, that speaks to the need to bring a whole community into this. I have another question from the audience as we start to think forward. So this mRNA vaccine platform, you described it as being kind of a plug and play system. What does that mean? And can you tell us about the potential of using this type of technology for other health reasons like cancer or HIV?
Yeah, I mean, this is the type of technology. This is a moment that we would call transforming vaccinology, where a technology has proven itself, obviously, and it's a type of technology where plug and play means you can just about utilize it towards any disease, obviously with the right types of experiments and all of these things, right? But Moderna actually started as a cancer therapeutics company. I think even still their largest portfolio of products is in the cancer therapeutic sphere. They have in clinical trials or going into clinical trials with flu vaccines, respiratory syncytial virus vaccines, CMV, all types of viruses. And they are, along with many other messenger RNA companies, really working in that direction of saying, this has a utility to help in so many spheres. So let's just get the job done. I'm hopeful that this is a moment where we will, number one, gain some trust around messenger RNA technologies, but then also, gain the types of resources that we need to investigate all these types of delivery, or this type of delivery platform for all types of therapeutics or vaccines.
With new variants of COVID spreading, what sort of work have you or others been doing on future coronavirus vaccines? And do you think we'll have a yearly vaccine with flu, or do you think there could be a universal vaccine?
So the variants are the reason why I am a year later not sleeping. Because so these variants, they call them variants of concern, but right now what it's looking like is that we're covered with the types of vaccines that we already have. As long as we can keep the mutations in the virus at bay, basically. There's certainly a space for universal coronavirus vaccines. I actually started at the VRC. I guess I started in coronaviruses and universal influenza, but I also have worked on universal influenza vaccines. And so this is a concept that is notable in other viral families as well. So whether it be changing the current vaccines to match circulating in new and novel strains, or coming up with vaccine technologies that are more universal and target multiple different strains at the same time, there's certainly different ways that you can go about that.
So question from the audience, to put a personal spin on this, have you been vaccinated?
Yes.
I have as well. Talene, I don't know if you're 16 yet, because some of the
I am not old enough yet. I'm 15.
Yes.
Oh no.
We're still working on it for you.
We're still working on it for you. But yeah, I've been fully vaccinated since January 25th.
So I actually just got my Johnson & Johnson vaccine on Saturday, which leads me to the next question, which is, what are your thoughts on what's happening with the J&J vaccine and the AstraZeneca vaccine in terms of some of the side effects that have been recently discovered?
My thoughts are that I'm very happy that we have a system that understands how to look at these types of issues very deeply, vets these products for their safety very deeply, pauses the rollout of the product shall they see a problem, but while they can investigate. So I'm very thankful to be here in the US where that is happening. At least as of yesterday, there were about 6 cases of blood clots that may be attributed to the Johnson & Johnson vaccines out of 6 million doses that had been given. So those numbers are very small. And compared to other medications, for example, birth control, or even COVID-19 has caused blood clots, right? So compared to that, those numbers are dismal, frankly. So I'm just happy that we have a system that investigates it. And I like to remind people to always follow the numbers. Have your concerns if you want, but remember to remain informed as the data continue to come out. And if you are not in the mental space to trust the regulation authorities, or like the FDA or the CDC, then at least find a space where you trust yourself to make an informed decision. Because the truth of the matter is, vaccination is always a choice. So making the informed decision about the vaccine that you want to take when you see, as you see the data, is the most important part.
Regarding concerns that people might have, can you help us understand the mechanism of how mRNA vaccines work? Like, can they change your DNA, and what's the difference between Moderna and Pfizer?
No, they cannot change your DNA. So messenger RNA sends a message to your body cells, kind of like a Snapchat message, a very quick message for your cells to make a spike protein. I was gifted these little things, which are crochet viruses. My goddaughters gifted these to me. But so this is the spike protein. It's on the surface of a coronavirus, these little red things. And this is what the virus uses to get into a cell. So if you can teach your body how to see this without getting infected, then you might be able to prevent an infection later. And so that's exactly what the messenger RNA vaccines are doing. They are expressing this spike protein on your cells transiently. So what that means is for a short period of time, so that your immune system can learn that spike protein in so much detail, so that when a virus comes your way, your body can defend itself against that virus. And that's how it works. Messenger RNA does not go into the compartment of the cell where your DNA is housed. So it's not interfering with your DNA at all. There's no way for it to change your DNA. Yeah.
No, I think that's always really helpful for people, again, to understand exactly what's happening inside your body as this vaccine gets to work. So another audience question here around the news. So the headlines today have been talking about some of these breakthrough infections of people who have been fully vaccinated, but have still developed severe symptoms and in some cases died. Now, you mentioned before about like looking at the numbers. Can you help us put these numbers into perspective?
I don't remember what the latest numbers were. I feel like I saw 70 somewhere, but I was not briefed on that this morning. But anyway, so yeah, the numbers are extremely small. And also breakthrough infections from a vaccine, in vaccinated people, is not something that is unexpected. We certainly would expect that, thinking about hundreds of millions of people who are being vaccinated all over the globe. To have a few hundred cases of infections that are serious infections is something that you might expect. Also remembering that humans are extremely diverse, right? So the more numbers of people that get vaccinated, the more you'll start to see more and more breakthrough infections. But it's not a cause for any concern, and it certainly does not speak to the efficacy of the vaccine. The efficacy of the vaccine remains steady at above 90% as the data in what I call the phase 4, which is the rollout of the vaccines, continues to roll out. So it's very clear that these vaccines are working, and it's unfortunate that there are breakthrough cases. I wish that there was a way, maybe the next generation of scientists can create vaccines that are completely 100% effective. But right now we are in this space of, 90-95% effective vaccines, and there are going to be breakthrough cases. And the most important part, especially as we haven't reached even close to herd immunity, so the point of where the population is substantially vaccinated and transmission is so low that you might not come in contact with the virus, is to remember that even as being a fully vaccinated person, you want to take heed to the recommendations of the CDC and remain masked, especially in around large crowds of people, and just take heed to what are your social responsibilities in a pandemic.
Looking more toward the future and going in with another audience question here, do you think it's possible or realistic for the coronavirus to be completely eradicated?
No, not in the near future. I think that what we have in our hands is a pretty steady state transmission. And even when we get to the point where we have, 80% of people vaccinated all over the globe, the virus is still going to exist, not just in people, right? There are still reservoirs of animals that this virus exists in, whether it be bats or whatever type of animal. And so most likely not eradicated, but we will get to the point where it's not a topic of conversation, where there is so little transmission that it becomes something like a seasonal influenza, for example, where yes, people get infected every year. And unfortunately, maybe some people will succumb to those infections, but it is not, we won't be in a place where it's at a pandemic scale.
And going on from that, what does it mean that some people have side effects to the vaccine and others don't? Does it mean that their immune system is not being activated to respond to the vaccine?
So I try not to call them side effects as much as possible and just call them effects, because you're right. It means that for people who have these effects, whether it be fever or chills or fatigue, which is what I had a lot of fatigue, particularly after my second dose. These are just the effects of your immune system waking up and getting to work. And so some people do not have as severe side effects. And anecdotally, particularly people of age, we don't necessarily know what the mechanism of that is, but we do know that those people are still as protected as the people who do have those other types of effects. And so we can't really say that if you don't have effects, then you're not going to be protected. My best friend was very concerned about that. She felt like she might've gotten like the placebo or something. And I'm like, no, it's fine. You're just lucky.
I can definitely attest to that generational difference in my family for sure. I was on the couch with the chills, right?
My grandmother never talks in our group chat, but there was about a week or two span where basically my entire family was getting vaccinated, and all of my cousins were like, oh my God, my arm is about to fall off. I'm having a fever, like all of these things. And my grandma's like, oh, I didn't feel anything. It's like her first time ever speaking in the group chat. She was bragging about it. So it's just person to person variability.
All right, so we're going to shift gears for our last few questions here to really listen to your vision for the future. Where do you want to focus your energy to have the greatest impact?
Well, I think probably I'm going to have to focus my energy on a break as soon as I can, to really just kind of wrap my brain around what is still a very long career trajectory for myself. So that'll be first. And I will be starting a lab somewhere that is currently undisclosed, at a place that I think will really help me to move my research aims in the direction that I want. So I will remain invested in pandemic preparedness. I will definitely remain invested in viral immunology for pandemic preparedness, whether it be for coronaviruses or other respiratory viruses.
And then a last question for the audience here. People have been just fascinated by hearing your story and your work. And I think this is a lovely question. How do you think about paying it forward?
Oh, in so many ways. I just sent off an email to get an endowment at my university. So many ways. That way, the resources that I will gather over my lifetime will certainly be used to give back to the community as they always have been. I make it a particular point to make sure that I mentor students that most people probably wouldn't even look at their resume because they have lesser experience. And so I started in high school. And so I tried to take onto my team, not in the last couple of years, because it's just been so difficult with the pandemic and also my career transition, but students from high school into the lab, paying it forward in that way, continuing to talk to the community and making sure that my science remains relatable to everyone is extremely important to me. And yeah, social justice. I actually have made it a point that I stepped into this space that is such a privilege to be asked by journal editors to write perspectives for like nature. I never would have thought that I would be in that place, but they're asking me to write about things that are science, and I'm saying no, but I'll give you, a document about how my science and social justice kind of intertwine with each other. And so paying it forward in that way and being very visible on and remaining active on as many committees as I can, whether they be the diversity committee at the Vaccine Research Center or education committees at community health centers and all of these things.
I just want to say COVID vaccines have given us a tool for tackling the biggest challenge of this pandemic. Thinking about 10, 20 years in the future, what new challenges do you see emerging for both science and our society, and how do we tackle those?
I think one of my hopes is that one of the challenges that we will not be dealing with, although I'm afraid that we still will be, are major social justice issues. So when you have just minoritized communities and injustices all across the board, it's going to trickle into any workspace and into any career, science included. So I think that one of our major challenges, and I'm very happy that the CDC director and the NIH director are really stepping up to the plate with that, are racial inequities and injustices. So that's one of our major challenges. And then secondly, I think that this moment has shown us that resources in understudied viruses or other scientific fields are so needed. And I think that I forget what we estimated, but someone can look it up. It's written in a perspective that we wrote for Journal of Clinical Immunology. But there are billions of dollars that will have to be spent in order for us to really prepare this adequately, or actually better, because we could have been better prepared even for the other 23 viruses that are necessary for us to be prepared for. And so we're going to have to learn over the next 20 years how to strategically allocate the resources, because the billions exist. So we know that. It's just about where we spend them. And that's going to be a very important question, I think. And then the other thing too, is that I get a lot of, how can people follow in your footsteps and how can people be like you? And to remind the younger generation that I am only one person and that I am human and that we all are, and that being the best you is most important. And if the best you is better than me, by all means do that. And if the footsteps that I have laid out don't fit your purpose, bask in what is your purpose is most important. And then from where I will be sitting in 20 years, hopefully in a place of leadership at some amazing institution or even my own, is reminding the people that are at the same leadership table as me is that everyone's purpose is different and that we need to really embrace every single person in order for us to get through all of the plethora of biomedical problems that we have.
That's such a perfect note to end on. Thank you, Dr. Corbett, for this really fascinating conversation. I think you've helped us, really appreciate tonight, that science doesn't exist in a vacuum, right? It's this part of this complex world and the systems that we interact with every day. And so, I think one of the lessons that I take away here is how your work is this model for how science and scientists too can really embrace that complexity. And that's so important, especially for young people to see as they're looking to see how they can change the world that they're living in for the better. And so we are thrilled to welcome you into the Franklin Institute's family of laureates, who each embody our museum's mission to inspire that passion for learning about science and technology.
So thankful. Thank you guys for hosting this.
So for those of you who enjoyed our program tonight, we also welcome you to join us next Thursday for a conversation with another one of our 2021 laureates, Dr. Monica Turner, who'll be in a conversation about forest fires and the future of our climate. So another grand challenge facing our world. And of course, all of our laureates will be receiving their medals in a virtual ceremony on April 29th, a ceremony that's made possible by the generous support of our presenting sponsor at Bank of America. So for more information on those events, you can check out our website at www.fi.edu. Dr. Corbett, thank you again for your incredible work and being such an incredible inspiration to so many young people out there. And thanks to all of you for joining us tonight. Please take care, stay safe, and we hope we'll see you at the Franklin Institute soon. Good night.
Bye.